Okay, hello everybody. Welcome to the August 21st back to school edition, well, almost back to school edition of Micro Seminar. We are very, very excited to have Mihai Pop presenting on metagenomics assembly today. He is at the University of Maryland in the Department of Computer Science, Center for Bioinformatics and Computational Biology. He did his bachelor's degree in Bucharest, Romania, a master's and a PhD at Johns Hopkins, and today I believe is going to help us all start to get up to date on the very, very grotesque complexities of metagenomics assembly. So without any further ado, Mihai, take it away.
Uh, thanks, uh, thanks, Cameron, and thanks, Jen, for organizing all this, and also thanks all of you who are watching this. I'm not kind of strange; I don't see you. So I'll start sharing my screen. So hopefully that will work. Um, there we go. Um, so what I want to talk to you about is metagenomic assembly, and I kind of realize I'm just diving in for those of you who don't know what metagenomics is or what assembly is. But the basic idea is that we can now sequence mixtures of bacteria and then take those mixtures, take the DNA sequences from those mixtures, put them together, and hope to reconstruct organisms or genes from those mixtures. And I'll give you a little more details about this. I apologize at the beginning, I'll probably go fairly fast, and hopefully it won't go too much over time. Let's see. Let's see. So the basic idea of sequencing in general, and also this applies to metagenomics, is that we start with a piece of DNA. That piece of DNA then gets duplicated. We essentially create multiple copies of it. Those copies get broken up into smaller pieces, and then those pieces are what get sequenced. So here on the screen, the little blue pieces are meant to represent segments that have been sequenced. And in those sequences, then we hope that we can take them, put them together, and reconstruct an original genome out of that. The reason we're doing this, and it seems to be like a very stupid process, but the reason we're doing this is because the sequencing technologies we have today have the sequences they can generate are much smaller than the sequence of genomes. So right now we're looking at the largest or the best technology that we have today is PacBio, Pacific Biosciences. It can generate reads about 20,000 base pairs in length. But if you look at the sizes of genomes of typical organisms, bacteria are in the range of 1 to 5 megabase pairs. Most eukaryotes are in the hundreds of megabase pairs to the gigabase pairs, and there are even larger organisms out there. So there's simply no way of actually taking the genome of an organism, shoving it into a sequencing instrument, and getting an assembly out of that. And to further kind of clarify what's going on, I'm taking this very nice metaphor for assembly, which is the Tale of Two Cities from Charles Dickens. The book starts with, "It was the age of wisdom, it was the age of foolishness." The question is, what would an assembler, what would a sequencing instrument do with this book? And you can think about this as having multiple copies of the book in a truck or something like that, and then the truck crashes, and you get lots of little pieces, and you're trying to put the book together. Right, so genome assembler, or what comes out of sequencer, will look like a collection of little pieces of DNA, the little pieces of the DNA or little pieces of the book that don't seem to have much of a connection with each other. So now we're trying to reconstruct this book. And of course, the way you would do it, and if any of you tried to do it right now, you take some piece from this mixture, right? Imagine you're going to the bin of a shredder and just taking pieces and try to reconstruct some document. You'll take one piece out and then say, well, you know, what can I add to this piece? And what you're going to add is something that overlaps it the best. So essentially, I'm going to look for some other piece that shares a lot of the sequence with the original piece. So in this case, I started with was the best, and then was the best of comes next. and I can keep this process, and slowly it looks like I'm reconstructing the tale of two cities. So everything looks great. And I think most of you will actually come up with this algorithm for solving the problem. And this is actually what people did originally. It's an algorithm called the greedy algorithm, because you always make the best choice possible at a particular time. And this greedy algorithm was the basis of many popular assemblers in the early days of genome assembly, including FRAP, which was the assembler, was the workhorse of the Human Genome Project, and Tiger Assembler, which was the assembler used to reconstruct the genome of the first bacterium ever sequenced. But one thing that's not perhaps immediately obvious is that this greedy approach can get stuck. So assume I've reconstructed was the best of times, it was the, and I'm trying to figure out what comes next. And well, pretty clearly I can put it was the age here, right? It was the age fits really well with the text that I had there before. But I could also put it was the best, or it was the worst. Now the question is, which one of these is it? And I have little information to go by at this point to try to reconstruct the book. And this problem with the greedy algorithm has led to a number of algorithms that try to look at the problem in a more holistic way. So, and the basic idea of these algorithms are that we're trying to represent in a picture, in a graph, or a network, the relationship between the pieces of the book, or the fragments of DNA, and try to understand how they kind of go together globally. So the same information I had before, and I would call these overlaps between the different sequences, was the best, overlaps was the best of, and so on. I would draw a little arrow indicating that was the best, overlaps was the best. Hello? Okay. I heard some noise. I'm not sure I'm supposed to do anything. Let me know if anything goes wrong, and I'll come back. Okay, so this is the situation that we have, and you can see that this ambiguity, when I get to times it was the, and I could have either it was the age or it was the worst coming out, is pretty clearly represented in this picture. Now, why is this helpful? Why is this more helpful than the greedy algorithm? The reason is that I can ask some questions that are more global than just simply saying what's the next thing that comes along. I can say, well, if I want to traverse this graph to reconstruct the book, then it's pretty clear that I have to, when I get to times it was the, I have to go to it was the worst of times. Well, why do I have to do that? Because if I follow the path to the graph from times it was the to it was the age, there's this whole section of the graph that I'll never be able to glue together to the book. So it's kind of it's sort of like taking something apart, and when you put it back together, there are pieces left over, right? That's we don't want to get into the situation. So just looking at this structure, it's pretty clear that I can just go to the, I have to follow it was the worst of times, and so on, and then get to it was the age. So actually reconstructing the book correctly. And these approaches, these graph-based approaches, are what is commonly used today in genome assembly. Of course, I am kind of, for pedagogical reasons, I am kind of tricking you a little bit. And the reality that even using these graph-based approaches, we can get stuck. So I can build a graph of what the book looks like to a genome assembler. And if I just use these words of length 4, like I did before, the segments of 4 words in length like before, I could easily look through this graph and reconstruct it was the best of times, it was the worst of times, it was the age of wisdom, it was the age of foolishness, which is the correct reconstruction. But I could also easily reconstruct it was the best of times, it was the age of wisdom, it was the age of foolishness, it was the worst of times. Right? They're both words or both phrases, both sentences fit with this structure very well, and I don't know which one of them is correct. And this is particularly, I mean, you should think a little bit about this, but essentially both of them are grammatically correct in terms of English. It's just that one of them was what Dickens chose, and the other one is what we reconstruct here. So just knowing English will not tell us which one is correct. And the same thing is true for biological data. If I'm going to look at an assembly of a genome, and just a string of letters A, C, G, T, I will not know if that string is a correct string or an incorrect string just by staring at sequences. So if the assembler makes a mistake, it's very hard for me to figure out that that mistake is there. And just to kind of bring this away from Dickens and to biology, this is a picture of what an assembler sees if it's trying to reconstruct a fairly simple bacterial genome called Mycoplasma genitalium. And it should be pretty clear that just by staring at a picture, even by using fancy computer algorithms, you cannot figure out the sequence of this organism. So, okay, so I like what I used to say, that the metagenome or your genome assembly is impossible, and in part because I am in the situation where I can reconstruct many different genomes from the same exact data, and I have no way of knowing which one of them is correct. But the thing is, it's a little more nuanced than that. Essentially, what I'm looking at is it's a problem of information, right? And I like to think of the whole biological process as kind of like a leaky hose, where I start with some organism that's sequencing, it generates a set of reads, they get assembled, and I get an assembly. And at each step in this process, in my hose that carries information from the genome to the final assembly, that there are a bunch of leaks. So sequencing process loses information, the assembler might lose information, and so on. And what I'm really hoping is that there's at least a drop of water coming out of the other end. The question for me as a computer scientist is really what is the information that gets lost, and what is the information we can retain throughout this process, so we can hope to get useful information on the other end. Okay, so one thing that should be clear is that if I had longer pieces of DNA that I reconstruct, I should be able to do a better job. So if I'm looking at the same picture that I showed you before, where I was very confused about whether I would have it was the beige of the best of times was the worst of times, it was the age of wisdom, or if it was the best of times, worst of times, or was age of foolishness, or something like that. I couldn't tell that if I just used words of length or strings of length of 4 words in length. If I have these longer words that are now that are now 6 words in length, I can immediately figure out exactly what the correct string is, because as soon as I see it was the best of times it, I know that I have to start here, follow it was the best of times it, and so I'm essentially resolving this ambiguity coming out from it was the in 3 different directions. So the assembly becomes much easier. And again, to bring it back to biology, here's an assembly graph generated from the E. coli genome for read length of about 50. Once you go to 1,000 base pairs, the graph becomes much simpler, and by 5,000 base pairs, the graph ends up being like a simple circle. So that's essentially the whole E. coli genome correctly reconstructed by the assembler. And you can show mathematically that assembly can fall. The relation between repeat length and the length of the reads is really what drives a lot of the complexity of assembly. And you fall into roughly three broad categories. When reads are very long with respect to repeats, then anybody can write an assembler that works correctly and well. If reads are much shorter than repeats, you get in this situation where I can reconstruct a genome, but I can reconstruct an exponential number of genomes, all of which seem to fit the data equally well, and I don't really know which one of those is the correct one. In between, it's a kind of strange situation that computer scientists love to talk about, which is this place. This category of problems called NP-hard, where essentially I know there's a single answer, so the problem is not undetermined, but finding it would require me to try all possible ways of putting the genome back together. But this essentially brings me up to the question of what repeats might be. So if I'm looking at a single genome, the repeats are usually drew them in green. And there's some segments of this genome that are repeated in multiple copies throughout the genome. So you have different, this green piece is repeated in multiple places. In typical bacterial genomes, repeats are fairly small. Some of the larger ones is the ribosomal RNA operon, which is about 6,000 base pairs in length. So if you think about, if I'm looking at a single bacterial genome, if I have PacBio data that can span over 10 to 20 kb in length, then these repeats are not the problem. They're much shorter than a read length, therefore the genome can be assembled in one piece. And there's a fair amount of work demonstrating that that's true in practice. In the case of a metagenome, where I have a mixture of organisms all put together in the same sample, and I'm trying to reconstruct individual organisms, I can run into these more subtle situations where I have different strains of the same organism that are virtually identical. So I'm showing this with green parts, are essentially identical across all these genomes, but they differ in subtle ways, whether there might be an antibiotic resistance gene that's present in one of them, or there might be a phage insertion in another one, and so on. And those small differences are the only thing that differentiate between these different strains of these organisms. In that case, clearly the repeats length is much longer than what read lengths we can generate today. So we might expect now the repeats to be in the hundreds or thousands of base pairs, and to a point where there are no technologies right now or in the near future that will be able to generate reads that long. So in metagenomic samples, we still have a big difficulty, even if we were able to generate more or fancier sequencing data. And just to demonstrate that this is really happening, I'm not just making that up. Here on the top left, there's a picture of 4 Pseudomonas aeruginosa strains, and the different colors indicate blocks that are identical between the genomes, and you can kind of see that the strain at the top has a whole region flipped with respect to the other ones below, and then the different strains have insertions or deletions of in different places in their genomes, but the blocks themselves are very similar. And also on the right, there's a demonstration that these are 2 strains of an organism called Citrobacter that was identified in the gut of an infant. So these are the strains that have differences between them. You can see where the empty blocks are in this circle picture, and these so this genome, not only are they different, but they coexist in a sample, and they have a different behavior across time in the gut of this infant. So so metagenomic assembler that wants to separate these out and understand their dynamics will really have to do really has a really challenging task ahead of it, because it has 2 virtually identical genomes with small differences between them, and has to separate them out. At the same time, I think there are a lot of questions biologists are asking that really need that separation, and there are some examples of questions here. I won't really dwell on them. Okay, so the first thing people do, so what I've told you so far is that repeats are a big issue, right, in metagenomic data and genomic data, and that's what affects genome assemblies. So one very common approach to dealing with repeats is essentially saying, well, I'll just get rid of them. I understand there's an ambiguity. I know I can't solve that ambiguity, but if I remove the repeats, at least the segments of the DNA that I reconstruct are going to be correct. I won't get the complete genome, but the pieces that I'm going to get are correct. Right. So in our case, in the case of Dickens, actually I'm showing you more of the book than the first paragraph. And here it's pretty clear it was the repeat that really confused our reconstruction, and it occurs many places in the book. These are just a few of them that we have. So the question is, well, how will an assembler figure out what this string is, and then use this string to try and eliminate it from the book, so then I can reconstruct just parts of the book that don't contain the repeat. And if you do simple counting, you can figure out very quickly that it was the repeat, because in what I'm showing you on the screen, it was there occur 17 times But the next string of 3 words that occurs in this text only occurs 2 times. So there's this huge discrepancy in the number of copies that I see the string of words, and because of that, I can immediately tell you I know there is a repeat, and I can eliminate it. Right? In the case of metagenomes, this no longer really works. And part of the reason is that there are organisms that are very highly abundant and organisms that have very low abundance in a metagenomic sample. And if I'm just looking at how many times something occurs, I might mark everything in the most abundant organisms being a repeat because it occurs much more frequently than the average of the rest. And we have worked on a variety of approaches that use other ways of figuring out what repeats are, using, for example, graph-based techniques developed originally in social network analysis to decide that particular node in the graph is a repeat because it's the other thing.
Mihai, can you hear me?
Yes, I can hear you.
I'm sorry to interrupt you. We lost sound on you for a second. Can you go back and start that slide over?
Which slide you lost?
Just the current one that you're on.
Oh, the current one. Okay, yeah. Okay. Is it okay now?
It seems to be, yeah.
Okay, sorry. One thing that keeps coming up and people have been asking me about this is when I've been giving talks like this is, well, can mate pairs help? And because we can generate sequencing data that provides additional information than just what's contained in the reads. And specifically, this type of information that we're getting is that I have two reads or pairs of reads that are connected to each other because we know how far apart those reads are. So if you remember on the first slide about sequencing, shotgun sequencing, I had little blue pieces at the ends of DNA fragments. Simply knowing that those 2 pieces belong to the same DNA fragment now constrains the assembly. In effect, if I have a link between these 2 blue nodes in an assembly graph, telling me that they are at a certain distance apart, I can look at the assembly graph and ask, well, there are 2 different ways of getting between these 2 blue nodes. One goes to the little circle, and the other one goes to the oval. Well, the circle and the oval are different lengths, and if I know the length between the blue nodes, then that will allow me to choose the path on top that I thickened versus the one on the bottom. Thereby, I'm resolving these repeats, this ambiguity in the graph because of these nodes that have multiple ways to go in and multiple ways to come out. Okay. And we've tried doing this for single genomes and seeing how well it works. And the somewhat surprising result was that this approach doesn't work very well. So here's a representation where we generated many different pairs of reads from a genome, and try to see how much of the genome we can reconstruct correctly, essentially how much of the repeats are resolved. So if I get to 100%, that would mean the genome is reconstructed in a single piece. And it's pretty clear that it doesn't matter how much data I generate with mate pairs, and it also doesn't matter which size it is, because I started with size 300 going up to 35,000, which is this green line here. In all cases, I stop short of actually resolving the genome completely. So the information contained in mate pairs is not entirely sufficient to solving repeat information. And part of the reason why that is the case is because there are many paths in the graph that have the same length. So if I'm looking again at these red nodes that were in the previous slide marked in blue, you can see that now there are 2 paths in between them like before, but now those 2 paths have the same size. They're both drawn as a circle. So there's no way, by just knowing how far apart the 2 red nodes are, whether I should choose the path going to the white node or the path going to the green node. So that's one of the challenges we're facing. And ideally, what you really want is for every repeat in the genome, you would want a pair of reads that exactly span that repeat. So in the case that if I have this green pair of nodes, I know I have to go from green node on the left to the green node on the right, and therefore I can make this connection between them, and I know which edges I should take for this repeat. Of course, I don't have the luxury to figure out what all the repeats are and generate mate pair libraries for every single repeat. So some mate pairs are not quite as useful as they at least appear in theory. Um, the other thing I want to mention, because when we're looking at metagenomic data, so I mentioned one of the challenges that we have with metagenomic data is that if I have a wide variance in the depth of coverage between genomes, the genomes that are very high coverage might appear to us to look like repeats. But there's another part of this that actually it's a little less obvious, which is that if I'm looking at the genomes that are not very well covered, if I don't generate enough reads from a genome to fully cover that genome, then I might actually make mistakes. So here's an example of what kind of mistakes I might make. I might have the same graph that I had before, but in this case we're missing information because I just didn't have enough reads to tell me that it was the best is present in my text. So in that case, I have a graph, and when I'm looking at that graph, the graph looks simpler than before. So the best is not there, wisdom is not there, and the structure of the graph is fairly simple, and it tells me immediately, well, I know what to say. It was the worst of times was the age of foolishness, or it was the age of foolishness was the worst of times, but there's no best or no wisdom. And the reconstruction that I'm coming up with is something that's not in the book. It's something that's incorrect. And yet the graph doesn't tell me that I have alternatives. So in this case, because I don't have enough information, I'm making mistakes, and I do not know that I'm making a mistake. And this is something that does not really occur in single genomes because we can generate enough sequence data that this situation doesn't occur. But in metagenomic samples, when the low abundance organisms are probably going to be incorrectly reconstructed. So I'm coming back to this idea that, well, metagenomic assembly probably is impossible. So in metagenomics, separating up strains will not work even if reads are very long, because we can't generate reads as long as genomes nowadays. Mate pair information is not very, very useful. And errors are really impossible to avoid, especially when you're looking at these places where there's missing information. And then to cap it all, a lot of the problems related to assembly are computationally very difficult to do. So you need large machines to solve this problem. You need lots of cores, and oftentimes you need to run the assembler for a long time in order to get a good answer. The question is, why would anyone bother with assembly then? So one thing you can think of assembly as simply being a way of compressing data. So this is an example where we took a sample from the Human Microbiome Project. This is a stool sample. We had about 11 gigabase pairs of sequence going into it. And after we assembled the data, we had something like 194 megabase pairs of data. So we had part of 174 megabase pairs in the assembled data and about 20 megabase pairs unassembled reads. And then we took the same exact data. So we took reads, we shoved it to the assembler and then did the analysis, and also took the reads and passed them through the same analysis but separately. And the analysis in this case was run with a tool called metafiler that's trying to estimate the taxonomic composition of the sample. And the results were essentially the same for both approaches. The only difference being that running the reads is 11 gigabase pairs through metafiler took much, much longer than taking the same reads, assembling them, and then running metafiler on the resulting assembly. So essentially, computationally, I can do things much faster because I've compressed the data through the assembly process. And this makes a lot of intuitive sense. If I have an organism that's present 10,000 times in my sample, there's no point in analyzing every single gene in that organism 10,000 times. You should only analyze them once, and assembly helps you do that. The other part with assembly is that we do need assembly. We do need to reconstruct genomes, and we can't simply rely on us saying, I'm going to take the reads and align them to a database, because there are many, many things in many organisms in real samples that we have never sequenced. We have never seen their genes. Every day you see new papers coming up, finding new organisms that have kind of strange behaviors or strange gene complements and so on. The only way we'll get them is by doing sequencing and assembly of these samples. We also have an interesting situation where we have marker gene surveys of samples that reveal interesting phenomena, interesting organism or strains of organisms that might correlate with the clinical parameter. And now the question is, well, what's the organism? What are the genes in that organisms? And again, the only way to get to those is assembly. Right? So assembly is absolutely necessary. And the other thing to say about assembly is I'm telling you, that there are many issues with assembly to start with, but there's some things that actually work. So this idea that repeats confuse us is certainly a problem, but it turns out not to be such a big problem within genes. So if you're looking at this plot, it shows you what percent of the genes in an organism can be assembled correctly. That means there's no repeat inside of them that confuses the reconstruction. And this is the percentage of genomes for which that percentage of genes can be reconstructed. So you can see that even for reads as small as 25 base pairs in this case, almost all the genomes have about 85% of the genes can be correctly reconstructed. Once you go to something like 1,000 base pairs, which is the top line, virtually all the genes are correctly reconstructed in bacterial genomes. And this is even though we cannot really separate strains or reconstruct the bigger picture in the genome, but at least the genes themselves can be constructed in single correct pieces that then can form the basis for analysis of the data. But now, a very important part that's oftentimes missed in these analyses is the fact that we do there are lots of problems, computational problems, and lots of missing data when we reconstruct genomes, and we have no way of knowing whether the result is correct. And so the figuring out whether this result of an assembly is correct is really critical before we trust the data to do any analysis on it. I would like to say, this is something, it's a sort of controversial topic at some point, but this was very well understood in the early days of genomics. And if you read the first paper, the first bacterial genome paper of Haemophilus influenza, you will see there's a huge part of the paper that's devoted to how it was validated that that's the actual sequence of the Haemophilus influenza genome. And at some point, we kind of started to trust technology too much. And there were recently a couple of papers on, for example, the assemblathon competition, and people said, well, it's great we did this because now people understand genome assemblers make mistakes. And like, well, people already understood that assemblers make mistakes, and we just chose to forget it over time. And now we're rediscovering this concept. That being said, right, the big question is, how do we really figure out that the genome assembler produces the correct output? And first, you have to understand a little bit of where these errors come from. So some of the errors, you can have this idea of chimeric contigs of scaffolds, where we might have pieces of different organisms glued together and so on. You could have the genome rearranged, for example, and some of those errors might be unavoidable just because of because of the information provided to the assembler. Another type of error at some level is the fact that I have not been able to reconstruct the genome as completely as possible given the data. You might not see it necessarily as an error, but if I have a fragmented assembly that I could have made less fragmented, that I would consider an error. But these two types of errors are things that are inherent to the algorithms I'm using and the data that I am provided with. But the other part that's a little bit ignored nowadays in our community is this idea that software contains bugs. So software engineers believe a number of somewhere between 15 and 50 bugs per 1,000 lines of code. Your typical genome assembler has hundreds of thousands of lines of code, so you can imagine how many software bugs are introduced there. And I would like to say that this number of 15 to 50 bugs per 1,000 lines of code that relates to the work of a professional software engineer. So it's not even the fact that a lot of the assembly code is written by postdocs and this is essentially people without a formal training in software development. So the number of software mistakes can be even larger. So the question is, how do we really figure out when I'm looking at an assembly that the assembly is correct, given that I know that there are a lot of problems with both the data provided to the assembler and with the software that implements the algorithms. Okay, so most of the time what people do is say, well, I'm going to compare different assemblies and decide which one's better just by contiguity. So essentially, if you think about it, the only thing people are worried about is this missing information. The assembly that's more contiguous must be more correct somehow. The assembly is better because it's figured out how to put things together better. Okay, so but part of the problem with this is that this story, the contiguity is only part of the story. We really also want to find out the errors. Also, as an aside, I want to point out that there's a measure many people use, which is called N50. Briefly, what the N50 size means, it says that any contig larger than, if you add up all the contigs larger than an N50 size, their size will add up to half the genome size. So perhaps it's a little easier to see on these figures where I took here on the right, I have a multiple assemblies of the same data set using different assemblers, and I'm essentially starting from the largest contigs to the smallest, and on the x-axis is the size of the contig, and on the y-axis is how much of the genome is reconstructing genomes larger than that value. So if I take all the genomes, all the contigs larger than 60,000 base pairs in this case, then I'm going to cover this amount of, say, this green assembly. Right? So the N50 size is simply drawing a line to this curve at exactly half the genome size. So the green assembly adds up to this point, and then the N50 size is simply half of that level, and the N50 would be the number you see here on the x-axis. Okay, so it should be clear that if I'm comparing the red assembly with the green assembly, using N50, one would be defined by the red line here, and the green line here is not a very good measure. Essentially, I'm going to say, they're not exactly the same thing. I can't really compare them because the total size of the assembly is so different between those two assemblies. And this is true for one sample. It's very much true if I'm looking at multiple different samples with different characteristics, where in this case, the green assembly adds up to almost what 20 megabase pairs, 200 megabase pairs of sequence, whereas the blue one here is only about the megabase of sequence that I have from the assembly. So I can't simply compare things on a single number like an N50. I think curves like this are a little more useful because just looking at the curve, I can see that the green assembly seems to be doing better. It has larger contigs, first of all, and then it still has larger contigs up to a particular given point. So I can get to this point of 2 megabase pairs much quicker using the green assembly than I can with the red assembly. Okay. So beyond this, if I want to look at errors, I can actually look at just the assembly itself and try to find inconsistencies in the data. So one type of information I mentioned are these mate pairs. So the mate pairs give me constraints about how far apart the reads should be from each other. So in this case, I have here in a correct assembly, I have a 2-copy repeat. I know that I have a library with short mate pairs, a library with long mate pairs, and this is what they're supposed to look like. If the repeat is collapsed, so if the assembler somehow puts together these 2 copies together, then this outer mate pair becomes shorter than I would have expected, so I can see that as a mistake. And also these reads now no longer point in the right direction with respect to each other and are not connected properly. So immediately I can tell just by looking at the signature of how the reads and the mate pairs map to the assembly, I can tell you that there's a mistake there. And there are a couple other examples of this. I can also have situations that highlight errors where I can have a collection of reads that all map to that don't fit into the assembly, but they map partially. So in this case, the end of the read match is here, and the beginning of the read match is here. So there's not the complete map of the mapping of the whole read, but the read it has this kind of split alignment to the genome, and this happens for multiple reads at the same time. And this is a situation, for example, that occurs where the read spanning the join between 2 copies of a repeat now have no place to go in the assembly because those 2 copies have been collapsed and that join no longer exists in the assembly. And I can also look at things like polymorphisms, where if I'm assembling a single genome, uh, then I should not see places where I have a mixture of reads that some reads say there's an A there, and some reads say there's a G. So that's a possible misassembly. So these kind of internal consistency errors can be used to decide whether an assembly is correct, and also to flag regions in the assembly where it can be a mistake. And these are very useful for then deciding when you're doing the analysis that you're not getting fooled by the you're not getting fooled by mistakes in the assembly when you're doing your analysis. There's a nice plot this is from Bud Mishra's lab called the feature response curve that kind of combines both contiguity that we talked about before in terms of trying to figure out how big are the contigues and also the number of errors. So here on the y-axis you have the total size of the assembly being reconstructed. On the x-axis how many errors the assembly contains of the types of errors that I just showed you. And then you can choose which assembly is better. For example, in this case, we'll choose this green assembly as being better because I can reconstruct more for any given number of errors. I can reconstruct more of the genome with the same number of errors. Or looking at it the other way, if I draw a line, a horizontal line through the graph, then essentially I'm saying, well, I can reconstruct 2 beige pairs with fewer errors than the other assembly. So that will tell me this green assembly seems to be better in this case. And this is, again, a more useful view of the assembly that's simply saying, hey, my assembly has 2,000 errors. Well, that doesn't tell me much, except that 2,000 errors, I can tell you green is above red, which means that green reconstructing more of the genome. Another another way of looking at the assembly, trying to understand if it's correct, is a really neat way of thinking about the problem. That's thinking in terms of probabilistic terms, where you can essentially you have some unknown genome, so we don't know what we reconstructed. We have a biological process to generate reads that I generally call magic. I call it magic because biologists mix clear liquids and expect something to come out of the and things come out of mixing clear liquids, which it's quite amazing. But also, and then I have the reads. The only thing I know about the actual genome is a set of reads. I don't know the genome. Then I'm taking the assembler and I'm constructing a genome assembly. And to know whether this genome assembly is correct, I can ask if I have some model of how biological sequencing process works. Then I can ask, well, is this assembly consistent with a set of reads? And I can ask this in a probabilistic setting, so I can get a likelihood score. And this approach is actually quite popular now that it was originally proposed as the way to do genome assembly, essentially optimizing this likelihood that the set of reads is consistent with the assembly is what Gene Myers proposed that the goal of the assembly should be. And it just happens to be computationally very difficult to do that, so we never. It was never really used in practice until fairly recently, when a number of papers, both metagenomic and also mixtures of viruses, are using these ideas to separate out different strains of an organism. And also, there's been a fair amount of work in this area recently in developing likelihood-based measures for deciding whether assemblies are correct, or more precisely for comparing different assemblies. And we have a software package that integrates all of this together, the likelihood score, and also these individual signatures of misassembly within genomes. But I want to get back and argue that so sorry, my computer keeps messing up. I want to show you a little bit of what comes out of this misassembly detection, and kind of, when I'm saying the devil is in the details, this is where we end up not really knowing what's going on. So in this case, I'm plotting the mapping of reads to a contig reconstructed from a metagenomic sample. And you can see there's this region in the contig that has high depth of coverage, and here these lines and these marks on the bottom tell you what our tool told us is wrong with the assembly. These are regions that our tool said, well, I think there's something wrong in the assembly in this region. So it's pretty clear that the tool correctly identifies some region in the assembly that's unusual. So that's possibly a mistake, right? If I'm looking at this, I can easily say, well, there's a misassembly, and I should be careful when I analyze the data. And this is really cool, because if I were to use a reference-based approach, to say if I knew the genome I'm assembling, the assembler makes this mistake where it takes these two copies of a repeat, puts them together. That's where you get the high depth of coverage. But if I take this piece of DNA that consists of these segments and I align it to the genome, everything will look fine, okay? Because the alignment, I have this segment, I have the blue segment followed by another unique segment here. That there's nothing wrong with the assembly. So I might not see, but just by mapping the contact to a reference, I might not flag this as a mistake. But I also find out that the opposite can happen, where the de novo validation, where my assembly is correct, I only have one copy of this repeat in the right place in the genome, and yes, the reference-based classifier, the reference-based validation says there's no mistake. In our case, the de novo approach might actually say there's a mistake simply because there are a bunch of reads that come from other places that simply get mapped to that particular region. And in that case, we would say, like in this example before, we would say here, I think there's a misassembly here, even though these reads probably are not from that genome, and the genome itself or the segment of the genome is correctly assembled. So it's a little tricky to really understand or to decide when errors are real errors, and this is, and I don't have a good answer for how to do it. You can also look at situations where there are true strain variants in the data. So here's a representation of an assembly graph, where there are two paths of the graph corresponding to two different genomes, the Citrobacter strains that I showed you earlier. And you can see that in the same place in an assembly, you might have differences between the reads and so on. But you also have regions in the assembly where the two genomes are assembled together, and a lot of these positions would be flagged as errors by some of these assembly validation tools, figuring out that whether they're errors or not require more careful analysis of the data. Okay. The other thing I want to mention is that there are a variety of approaches that have been developed to try to estimate the level of completeness of an assembly, essentially the HMP was looking at the number of core genes present in a genome. There's also a tool called CheckM that does a similar thing based on specific genes in the assembly. So you can look at the reconstructed genome from Metagenomics and say, oh, I think this is not complete because it only has 80% of the genes that I expect to be in a genome to be there. But even that kind of approach is not foolproof. And there's a recent paper from Jill Banfield's lab that essentially found organisms that are lacking certain genes that we consider core and commonly found in all bacteria. So especially as the reason why doing metagenomics is for us to look at previously unseen organisms. It's very hard for us to say, I know there's a mistake because this organism doesn't look like anything else we've seen before, because that might not be the mistake. There might be that it's amazing biological discovery that can put a bacterial genome in nature, which doesn't usually happen otherwise. So to end, I want to I have a few points that I'd like to leave you with. So first, I want you to be very, very aware of this principle, garbage in, garbage out. So this is a very common principle in computer science. And the basic idea is that the data you provide to the assembler has lost the information that's useful, necessary to reconstruct the genome. Then there's no way the assembler is going to reconstruct that information. So if you're using very short reads and you don't have mate pairs, don't expect too much of the assembler, because the assembler can only give you the results as good as the data was provided to it. The other part is that you have to be very careful about how you trust or how you validate the assembly, because ultimately depends on what questions you're really asking, and then a lot of times the errors or things that look like errors might be real biological signal. And I don't know of any single measure that I can say, well, measure this and it will tell you if the assembly is correct or not. None of those will be foolproof, and it really all depends on you very carefully looking at the data and analyzing it. So I think a key part is for you to kind of have a good idea before you delve into genome assembly and really figure out what you're trying to get out of the assembly. So if you just want to find genes, then you might use a different type of validation, and you also might have different metrics. So if I just want to find genes, I don't want necessarily really large contigs. I just want contigs to be correct, and so on. The last thing I want to say is that it's very important as we go forward that we start developing both validation tools and also standards. So, for example, a lot of the issues in validation and the analysis of metagenomic data arises from the fact that genome assemblers no longer tell you where the reads go in the assembly. So a lot of what we're doing is taking the reads, mapping them back to the assembly, and trying to infer from that things like depth of coverage or regions where there are too many reads lined up in the same place and so on. And a lot of times errors that we notice might simply look like an error because the alignment process that we used was suboptimal. And if the assemblers were able to tell you a depth of coverage for each contig based on what the assembler itself knows what it did, will be able to tell you which reads went and where in the assembly, then a lot of these kind of ambiguities caused by us trying to reverse engineer that information will probably go away, and our life would be made a lot easier. So that's all I wanted to cover. I do want to acknowledge a lot of people have contributed to some of the work both on assembly and assembly validation, and also I want to acknowledge our funders who have supported this work over the years. So thank you, and I guess I'll take questions to see how this goes. I haven't done this before. So.
Hey, well, okay, thank you very much, Mihai. That was very informative. You can undo your screen if you want to take questions in person.
Okay.
So anybody that's in the seminar, feel free to unmute yourself and speak up if you've got a question. Also, for those of you who are watching on YouTube or elsewhere, tweet questions to @microseminar or the #useminar. So I had a question right off the bat because I'm really excited. You know, this is sort of personally something I'm intrigued with right now and dealing with at this exact moment in my lab. Do you put up a website for a validation tool? Is that available right now?
Yeah, the tool is available.
Okay. And what kind of, so can you give some like sense of what kind of computational requirements are required to execute that?
So not a lot. So the biggest computational effort for the tool is essentially running an aligner.
Okay.
The rest I think is fairly straightforward. I haven't tried to see how it works on a laptop and stuff like that, and we're still kind of fine tuning.
Yeah.
But
Okay.
It works.
And what's the output for that tool? Is it sort of graphical based, or is it a series of like probabilities by location, or what are we talking about?
So it's not the graphical. It's not the graphical output. Essentially, we produce a collection of GFF files that essentially tell you which regions of the assembly contain errors.
Okay.
And we also we separately produce the likelihood, but the likelihood there's going to be a single number, right?
Yeah.
Then we also have the alignment of the reads in the assembly, so you can actually load that all into a genome viewer. So,
Okay.
So what I'm showing you, the graphical stuff I showed you is based on running IGV. So and you can load everything together.
Yeah.
But there's no fancier graphical output.
Right. Sure. Are there any other questions for Mihai?
So Mihai, I needed that validation tool like 5 years ago.
Oh, we had one back then, but it was a little different world.
Let's say, because we couldn't get any, we knew there were errors, but we couldn't get any validation tool to show it, except just the instinct of knowing that it had to be wrong. So, anyway, so thank you for having a new one, because we will probably be using it.
Yeah.
I wanted to follow up with you about your statement about standards. I was wondering if you could get into a little more detail about what you think the kind of standards should be for metagenomic assembly.
Yeah, so this is a tricky part. I mean, I'd like to get a better handle of what people really want to do with the output of the assemblers, but I think, I apologize, my camera jumps around as the mic don't. But back in the day, so the early genome assemblers told you exactly where every read went in the assembly and exactly how it aligned to the assembly. So you can think about the BAM file for every content. That's what the output of the assembler looks like. I don't see why current assemblers should not be able to give you that. I mean, I know why they're not. It's computationally expensive and doesn't quite fit into the brain graph assembly paradigm. The other thing that we don't have a good standard or a good understanding of how to represent the data is this idea that I'm not reconstructing a single genome, but at some level a pan-genome. And there are no good ways of showing that information or capturing that information. I mean, there are good ways. As a computer scientist, I know I know how to represent it in a way that makes sense to me. But the question is, how do you represent the information in a way that makes sense to biological researchers and that you can align things to and search against the database, that kind of stuff. So we don't have standards for that. And I think there have been some efforts trying to generate such standards, and that so far have fallen flat.
Yeah. Yeah, there's a lot of, I guess there's a lot of different needs out there, so it's going to make things a little more complicated.
Right.
And I just had a question following up on your comment about length isn't going to solve the problems. Yes. Because I thought, actually, I'm not sure if you were involved in it. I think maybe Adam Flippy was involved in it. There's some graphs coming out of Maryland talking about average repeat regions in bacterial genomes.
Right.
And the sort of moral of the story was that PacBio was getting long enough and good enough that you could get through that pretty quickly. And then with nanopore, I mean, I haven't... Again, we're all still new to nanopore, that this promise of longer reads seems to be on the horizon. And so, yes, right now everyone's still going for these shorter Illumina reads because of the volume and just the quality. But where do you see us like 5 years from now is my impression was that the length problem was probably going to get a lot better in terms of these long reads really helping with a lot of this situation.
Yeah, so my point is that for single genomes, for single bacterial genomes, it should work, right? So having reads of length about 20,000 base pairs, which is what we're looking at right now, and that's probably going to be the same nanopore or PacBio. It's going to solve repeats within the same genome. But if you want to, if you're asking, I'm looking at my sample, I have a mixture of 2 different organisms, or 3 different strains of the same organism, and that across time have different dynamics. Being able to separate those out will require some variant of haplotype phasing across much longer distances than 20 kb. And at that point, we won't have reads that go beyond 20 kb anytime soon. I mean, sequencing people might disagree with me, but even when you kill a bacterial cells, that the fragments of DNA that come out are in the order of several hundred kb. If you don't do anything with the DNA, that's the best you can do, right? So.
Well, our PacBio, our sequencing center says the biggest issue is not having people skilled enough to get high molecular weight DNA in terms of the length of the read. Like, that's what limits it, is the ability to get long DNA. So, but I've learned to never say never, because as soon as you say, like, oh, this will never happen, like, they'll come out with a new technology.
Yeah, I'm not saying that's never going to happen. I'm just saying that looking forward, I don't see this happening in the next 5 years. I don't see read length going much longer. And my guess is PacBio and nanopore will work really hard to lower error rates more than making the reads much longer.
So Cameron, did you see any questions come out on Twitter? Is anyone in the Hangout and want to speak up?
Yeah, I haven't seen anything coming through on the feed, but is there anybody in here? I mean, I know there are people in here. It's just whether you have questions.
Well, I will let you know, Mihai, that we had about, at the peak of it, there was about 30 people watching at once. And so, again, we are like really excited to be doing this in terms of just saving energy, saving time, and being efficient, and getting about the size of an average departmental audience. So, so thanks for giving a very popular talk. And I'm sure we'll watch the statistics go up. We'll probably have 100 views by tomorrow.
Yeah.
Great.
Cool. Yeah. Thank you very much for taking the time to share all this with us. I'm gonna ping you off the air one way or the other so I can get a hold of that tool. I want to start playing with it.
Okay. Thank you.
Okay.
All right.
All right.
Well, you can leave a minute open, but if not, we can turn off the live feed and